So thanks for your invitation and for this great opportunity. And I like to thank also Fatma Betül, who is who was a student in my lab, and now that I'm here to talk about some of the things that we do in at Bilkent. So just to give you a general idea about this cholinergic signaling, is that it is known as a neuronal pathway. And the way that I'm going to present here today is I'll tell you some biology, but then we use bioinformatics approaches to understand some of this biology and to come up with new hypotheses and to go on further. So cholinergic receptors are main part of the cholinergic signaling in neuronal cells. They're located on postsynaptic cells. And when we just like move, we are actually creating action potential. So what is happening is that there's acetylcholine in between presynaptic and postsynaptic cells. And that acetylcholine is directing the signaling through these muscarinic or nicotinic acetylcholine receptors. And something is happening such that uh, different different types of signaling occurs. So I'm going to have to stop just a second. I'm sorry, do you see me? I just need to go get some water. It seems like I'm having a little bit trouble with the talking. Just a second.
Okay, we're gonna wait.
I am back. I'm just going to need to share again. Sorry about this. I didn't realize I was going to be having this problem. Okay. So we talk. Can you hear now?
Yeah, we can hear you.
Okay, great. So as I mentioned to you, the neuronal pathways, similar things are happening in cancer cells. So acetylcholine is produced in one cell, it's going across another cell, or it is autocrine. It can affect on its own cell because in the cancer cells, you have many, many cells next to each other. They can exchange signals. And this signal transduction is happening through this nicotinic acetylcholine receptors, which I will talk to you about specifically. One of them is called A5. And what this nicotinic acetylcholine receptors do, along with some other muscarinic and adrenergic receptors, they take in calcium together with sodium, and they initiate a cascade of events such as activating AKT or MAPK and making the transcription factors go translocate into the nucleus and change the expression of many different molecules. So, and you might be familiar, AKT, MAPK, RAF, all of these are cancer-related molecules. They're signaling particles that are involved in cancer. So we decided to understand how this nicotinic acetylcholine receptors, their absence or presence of, affect these events in breast cancer cells because it hasn't been studied so much. And these cholinergic receptors, one of which that we're studying, A5, is found together with A3 and AB4. These are different types of subunits that come together, make up a pentameric form of the receptor. And this pentameric form is active. It is found on the membrane. It passes the membrane, and it allows this calcium to go in. And there are some homomeric function types as well. But A5, A3, and B4 are three different genes found side by side on chromosome 15. So we were interested if we lose this receptor, especially A5, which is known to be associated with lung cancer, what will happen in breast cancer? In fact, they have studied A5, one of the subunits that make up this receptor in gastric cancer, and they found that when they give nicotine, this AKT is just upregulated. It becomes functional, and it even results in cisplatin resistance. Cisplatin is a drug that is used in gastric cancer cells. So activating this A5, so getting this calcium and activating AKT is so much that cisplatin's effect on blocking AKT is not enough. So we were also interested in looking at if they have a drug resistance in these cells where we have or we don't have A5. So our aim is to develop a model in breast cancer with or without A5. And we did this, as I told you, that A5 is found on chromosome 15, and it has multiple exons, and exon 5 is alternatively spliced. That means some transcripts have it, some transcripts don't have it, but the main form has it. So we have designed or used three different siRNAs. siRNAs are those that bind to the mRNA and can degrade it or can prevent its translation. And there are five different isoforms of this A5, and we have used four of them and one general one. And we reduced this. So our model now is we have A5 depleted from the cell, and this was MCF7 cell. We could do this with three different siRNA molecules. And then that was mRNA. We looked at the mRNA and we reduced it. We looked at the protein. We reduced it by half at least. So we know that we have a model that depletes, not completely disappears, but there is still A5, but much less. So we did a microregion expression profiling. This was about 4 or 5 years ago. We did this with my student Shaika and Aisha, and we have analyzed the data. And we have identified, so what we did is we have siRNA treated and control cells. And we looked at what has changed, what kind of genes have been transcribed in their depletion or in the presence. And the A5 depletion increased P53 signaling, and A5 deletion decreased DNA replication and cell cycle. So it's actually acting like an anti-cancer molecule. So get rid of A5, it makes P53 signaling up, which is apoptotic, it could be, and cell cycle down. We did some qPCRs in three different cell lines, and we saw that MCF7 with P53 intact is more responsive. So this is cell cycle, this is P53, this is gap junction, and they're all affected in MCF7 compared to the P53 negative cells. So that means that A5 may have an anti-cancer agent, just like in gastric cancer, they have shown that A5 presence was not good for treatment. Its absence could be good for treatment. So this is our first finding, and I want to say for bioinformaticians that we have so much of this data now available. You're familiar in GEO, Gene Expression Omnibus. This is one of the data there, and there is like tons more. And it is possible to put things together and analyze, as I will show you in my next slide. So of course, when you do something as a biologist and a bioinformatician, you have to check what you have done is effective, right, in cells. So A5 depletion could be anti-proliferative. It could be good for treating cancer, but does it really reduce the cell number when you deplete A5? And it does. So we have identified that 50% reduction in cell viability. And the cells started to go into G1 arrest. This one shows that cells accumulated in G1 phase in cell cycle. And these pictures we take with fluorescent microscopy shows that BRDU, an analog of nucleotides, have been incorporated inside in normal cells that are expressing A5. But when you get rid of A5, cells that incorporate DNA molecules to make DNA becomes reduced. So DNA synthesis is inhibited. And this is my another student, Amira Yahya, who has shown that cell depletion of A5 has results in cell cycle. Next, then we decided to look at everything at the biochemical level because it is really important to see things at the mRNA level or at the cell level, but you really want to see things happening at the protein level. Because as you know, there's post-translational modifications and post-transcriptional modifications, and there is all kinds of different things going on after transcription. So what we did briefly, I'm going to show you, is that we have siRNA absent or present, and we have a biological replicate. And we saw that phosphorylated RB, which is one of the proteins that is reduced, phosphorylated RB is reduced when the cell cycle is arrested. So we see this. This is what we expected. And Casp7 is cleaved when there's apoptosis. So we see this again also whenever you deplete A5, cell cycle is reduced, apoptosis is increased, DNA damage response is abolished by reducing CHEK1, and DNA damage might have been induced. So this is what we have seen, which is good. So we are on the right track. We are continuing. So remember, I told you again, gastric cancer, when you have A5 and nicotine acting through A5, it prevents cisplatin act properly. So we said that if that is the case, and if we reduce A5, does it act synergistically with the doxorubicin or camptothecin? These are drugs that are used for cancer, doxorubicin and camptothecin. They inhibit topoisomerases that are important for DNA replication. So I just want to focus on this very part at the end so that doxorubicin itself, which is shown by black, reduces cell life, cell viability. So it is really effective. But if you use it together with siRNA, in the case of depletion of A5, it even reduces the cell viability more. So this shows that it synergizes with A5 doxorubicin and similarly camptothecin at a specific concentration. So all of these are suggesting that we are doing the right things. And we show again this in Western blot saying that doxorubicin increases apoptosis together with Si here. Okay, when there is a doxorubicin and you get a little bit higher of together with the siRNA, you have more cleavage of Casp7, you have more box, you have less CHEK1, and you have more H2AX. This is all showing you that there is an increased response comparison to Si alone or doxo alone. So this looks a little bit more complicated, but what you need to get here is that together with doxo or CPT, siRNA is more effective, which is good. So just to summarize this introduction, biological introduction, and which the rest of it will be a little bit more bioinformatics, is that A5 modulates cell cycle and drug sensitivity in breast cancer, which hasn't been shown before. It can be doing this through modulation of CHEK1 and TP53. And this is a tumor suppressor. This is a DNA damage inducer and regulator. So DNA damage response gene expression is affected by A5, which I haven't shown you, but we also found this out. And this study was currently further in study because we are checking the effect of TP53. How important we have to have a mutation and A5 depletion at the same time. So this was all in vitro and a little bit of microanalysis. So, but the important thing is that when you go to translational research, you have to really treat patients, right? If you have found something, if you have found a means to treat cancer, you have to test it in vivo. And how does that happen? So first you have to study ex vivo. Ex vivo means you take tissues and you do immunoprecipitation or you do cDNA, qPCR. You look at whether your gene is there or what does your gene do. And the second thing is that, of course, you can do in vivo studies with an animal model. But let me first talk about ex vivo. and how these breast cancer subtypes and prognosis could be important when it is concerned with A5 and estrogen receptor. Estrogen receptor is very important for breast cancer because it defines tumor subtypes. Some tumors have estrogen receptor positivity, they're known as luminal A, and some tumors lose estrogen receptor, but together with they lose progesterone receptor, and they don't have any HER2 amplification, which is ERB2. It's an EGFR receptor. So they're known as triple negative. These patients are generally not good prognostically. So that means when they're diagnosed, they may not survive as long as the luminal A, which has estrogen receptor positivity. You see this immunoprecipitation IHC slide. There is brown signal coming from the estrogen receptor, which is present in the tumor. But in ER negative patients, there is no estrogen receptor. Somehow they lost estrogen receptor and they lost their ability to signal together with estrogen receptor and estrogen, but they keep multiplying somehow without the estrogen receptor. So, an estrogen receptor, positive ones, multiply the tumor in the presence of estrogen. These can do this without the estrogen. And similarly, and there is a corresponding uh, like structure in the cell lines. MCF7 is here, relatively. 231 is here, more aggressive. So what did we decide to do is, what we decided is to check if A5 expression in cell line, does it depend on E2? Does estrogen receptor increases or decreases? This has never been studied, so we have studied this in different isoforms together with the PS2. This is one of the genes that respond to estrogen. We have studied whether there was an increase in estrogen treated cells in terms of A5. So, does estrogen induce A5? Yes, it does. In all of the isoforms, we have seen like fourfold. This is log level. That means at least fourfold induction, which is quite a lot, not as much as TFF1, which is the main primary target. Then we said that just one cell line is not enough. Let's look at the estrogen receptor negative cell line. We see increase in estrogen positive cell line in a dose independent manner. In every dose, it is induced A5 expression. But in MDA231 cells, which do not have estrogen receptor, it is not that much induced. And then we repeated this with different cell lines because it is not enough to use just a single cell line. So now what we have established is one connection between A5 and E2 nobody has done before, which is E2 induces A5. So this could be important. Why it can be important? So estrogen treatment induces cell cycle. Estrogen treatment combines with estrogen receptor induces cell cycle. Estrogen starvation, if you cut the estrogen from a cell line, it starves and it goes through cell cycle arrest. So we hypothesized that A5 depletion profile should be more correlated with starvation than with treatment induction, estrogen induction. So what we did in comparative transcriptomics, we have our own data and we have identified data sets in the literature, existing data set, when E2 was given and E2 was depleted. E2 is the estrogen here. And all in MCF7. So 3 data sets, we intersected them by using Venn diagrams. And what we have identified is that if a gene is downregulated by starvation, it is also likely to be downregulated by A5 depletion. So starvation and depletion are in the same direction, down, down, common genes, and those common genes were upregulated in estrogen. So this tells me there's this green region, 420 genes that are downregulated by estrogen starvation, downregulated by when I deplete A5, and upregulated if I put estrogen. So these are the genes that are kind of upregulated by estrogen but downregulated by these. And the opposite exists, that these 280 genes that are involved in FOXO signaling, etc., are upregulated by starvation, upregulated by A5 depletion, but downregulated by E2. So why did I do this bioinformatics comparative transcriptomics? To support my finding that estrogen negative cell lines like Cal51 does not respond to uh, estrogen in terms of increasing A5, but these do. So if I deplete A5, do I get something like a reduction of E2? So if there's no A5, is it going to be like starvation of E2 because estrogen is upregulated when there is E2? So I wanted to test this, and this actually proved to be quite significant. Why significant? I did a Fisher's exact test here, and Huma Shehwana is the student, PhD student, who has done many of these studies. And I have seen that estrogen treatment, everything was negative. Up, down, and up, down were more than up, up, and down, down. So this is a Fisher's exact test. It gives me an odds ratio. It shows me to show me that one thing is up, the other thing is down. Is this more obvious or is this more observed? And starvation, I have up, up, and down, down higher. The gene numbers here. These are the genes up in starvation, up in a depletion of A5. And I see that there is a signature of positive association with starvation, negative association with E2, given the odds ratio, which is highly significant at the p-value level. So I'm showing you this because it is very important to support your data, your results with statistics, and you have to do multiple statistics and representations. The next thing we have done is now that we have A5 mRNA, which is induced by E2, how is this happening? Is like estrogen receptor increased and A5 is increased, or is it just estrogen itself? So interestingly, when I look at the TCGA graph that is drawn here, TCGA is Cancer Genome Atlas. There are more than 10,000 patients and their mRNA protein methylation patterns, etc., and more than 1,000 breast cancer patients. So there are more than 1,000 dots here, each one showing a patient. If a patient has high estrogen receptor, it has it has a like a high for this cluster, there is really not that much of a relationship, but right here, if you look at, if the estrogen receptor is not there, you have higher A5 levels. So this shows like a negative correlation, although it is not perfectly linear. And what you see that they are negatively correlated. Estrogen receptor more, less A5 right here. Estrogen receptor less, more A5. So this goes a little bit off what we expected, right? We expected that estrogen receptor might be leading the A5 to increase when it is associated with E2, but that might not be the case. Maybe there is another protein that is induced by A5 as estrogen receptor that induces A5. So this is known as a secondary or tertiary signaling. So A5 may not be the first direct target of estrogen receptor, but it could be a target of what estrogen receptor induces, such as E2F1 or FOXA1 or some proliferative factor which induces A5. But this is all bioinformatics again. This is all data that is put into the TCGA, so you can get these figures easily. What we did is we have used more than like 40 tumors and some normals, and some of the tumors are ER negative, some of the tumors are ER positive. We checked their expression, A5 expression of these different isoforms, whether estrogen receptor negative patients have higher expression than estrogen receptor positive, just like we have seen here. Low estrogen receptor, high A5. Low estrogen receptor black, high A5. So this shows us really what we observed bioinformatically is what we see in the real qPCR as a validation. So I'm specifically showing this because it's very important to support your findings in bioinformatics with either ex vivo or in vivo that we have seen before. So one of the important things in our lab, what we do is a meta-analysis. Meta-analysis is another way to, to say things more confidently. And meta-analysis indicates that you get many data sets of the same kind that are outside in the geo or somewhere else. You put them together, statistically analyze, and to get a cohort that are similar, that are made up of similar studies, so that you have larger sample size, you have more confidence. So we have two cohorts here I want to show you. and I'm going to explain this figure in more detail. I have two cohorts that are collection of different data sets put together, and they're independent of each other. I call one of them ER cohort, the other one is survival cohort. So in this ER cohort, I have many breast cancer transcriptome data sets put together, and what I'm measuring is the ratio of a gene, expression ratio of a gene between ER negative over ER positive. So genes that are high in ER negative or genes that are low in ER negative. If you say 0, these genes are high. Each of these genes is high in ER negative patients, and all of these genes that are small dots here are low in ER negative patients, and A5 is one of them. A5 is high in ER negative. And what else I have here is something called PS score. PS score is I'm subsetting a group of genes that are induced by estrogen. These are induced by estrogen. I'm sorry, induced by estrogen. And I give a color saying that if they're induced a lot by estrogen, okay, in the absence of cyclohexamide, in the absence of cyclohexamide, I'm sorry, they are induced high in their secondary targets. I'm going to talk to you about this PS score now. So there's a compound called cyclohexamide. Cyclohexamide inhibits protein synthesis. So think of it this way. You have estrogen, it binds to receptor, it goes into the nucleus, it induces a gene. And to estrogen, binding to estrogen receptor, it induces a gene, doesn't require any new protein synthesis. But if a gene like A5 is induced not directly by estrogen receptor, by something that estrogen receptor induced, that means as a secondary target, first, what needs to happen? Estrogen binds to estrogen receptor, induces that X gene. That X gene needs to be transcribed, translated, go bind to the A5, and make A5. But if you inhibit translation, it cannot be induced, A5 or genes that are secondarily induced. So if you give estrogen alone, you're detecting both primary and secondary targets. If you're giving cyclohexamide together with estrogen, then you're detecting only those that are primary if there's no increase. If there's no protein synthesis, there is no A5. So although there's a little bit of an increase here, which suggests that there could be a mild primary target, A5 is mainly induced by another factor that is induced by estrogen. So what this tells me, this figure then, higher the PS score indicates that higher this difference between these two groups. So if estrogen is high, but it is dependent on protein synthesis, that's a secondary target. So high PS score shows you high secondary target potential. And all of these genes that are high in estrogen receptor negative uh, patients are induced by estrogen secondarily, just like A5. So this analysis made us assign A5 as a secondary target of estrogen receptor, which is induced by estrogen, but not through estrogen receptor itself, but through its secondary target. The next thing is that I am showing you this second cohort, survival cohort. This is another set of patients, but in these patients, we have measured a correlation between A5 and every other cell. This gene is upregulated in estrogen receptor negative patients and very much negatively correlated, downregulated. So this gene is lowly expressed in estrogen receptor negative patients and negatively correlated with A5. This gene is highly expressed in ER positive. It is positively correlated. That means A5 is one of these genes because it's very correlated with this set itself. These two are independent. So this is another thing I want to tell you in bioinformatics. If you're going to do meta-analysis, do independent cohorts. So you need to select your samples and split them into two different data sets. You estimate one parameter from one, another parameter from another, so that when you observe such a very high correlation, it is really due to complete independent sets, and it is more likely to be supported. Using the same data, ER minus over ER plus effect size, which means that This gene is downregulated in ER minus, this gene is upregulated in ER minus, given the value. And this gene is highly correlated with A5, and this gene is negatively correlated with A5, and these are all the genes. I can also color my data. Instead of PS score, I can color it by meta-HR. Meta-HR is a survival value, and it is log scale. What does a survival mean? It's an odds ratio. If it is greater than 1, and if it is greater than 1, and if it is 1, it is log is 0. If it is greater than 1, let's say 1, that means any gene that is this color, the brown, is 2 times more likely to cause bad prognosis. That means if you have that gene very highly expressed in your tumor, in the tumor, and that means that that tumor is likely to be a worse prognostically. So higher meta-HR scores, higher odds ratios, increasing the risk of death or length of survival, and good ones are increasing good prognosis, which means these patients having these genes upregulated instead of downregulated are surviving longer. Patients having these genes up survive longer, and these genes up survive less. So again, this correlation between ER cohort and A5 survival cohort is linear, and those genes that are associated with high ER negative patient expression also have bad risk or bad prognosis. So they're not good to have as high expression. So far, we have used these two data sets were microarray data sets. So they're transcriptomic data sets of breast cancer patients, but obtained from microarrays. Then we went on to TCGA, which is based on RNA expression, RNA sequencing data. And we have looked at principal component analysis. So we reduced the dimension of this data set, TCGA BRCA cohort, which is breast cancer patients that are made up of more than 1,000 tumors. And we have focused on primary and secondary targets of that we have discovered. These primary and secondary targets are going back to here. So these are secondary targets. All of these colored dots are secondary targets. We have identified them PS score. But we also identify the ChIP-seq based on the ChIP-seq data, chromatin immunoprecipitation sequencing data. Where does the estrogen receptor binds? Those are the primary targets, direct binding sites. So you can get that type of data from ChIP-seq. So we have primary targets from the ChIP-seq. We have secondary targets from the PS score. And each of these dots is a gene. And the expression of the gene from these 1,000 BRCA cohort patients is represented by a single dot here through a dimension reduction you might as well know as principal component analysis. This principal component analysis is telling you that there is a grouping of these primary and secondary targets. Primary targets seem to be on the left, secondary targets seem to be on the right, and there seems to be a distinction at the level of PC1. Even the PC1 is able to separate these two quite well. And when I color these genes, each of these genes now as secondary and primary, triangles and circles, are now colored by whether that gene is downregulated or upregulated by A5 depletion. When I look at this, secondary targets seem to be downregulated by A5. And just like A5 itself, A5 is downregulated as a secondary target. I downregulate secondary targets by the addition of siRNA. And many of the primary targets are upregulated. So again, you can see that A5 might be playing a role in splitting primary from secondary targets. And why this might be important? This might be important translationally. Why? Because this is the survival HR, remember the hazard ratio, odds ratio, like measurement. This says high risk, this says low risk for the survival. All the secondary targets have high risk. Many of the primary targets are low risk. So it is, in fact, I'm seeing here in another data set yet. This was microarray. Now this is RNA sequencing data from TCGA, and I see the same split. So I am seeing this in 3 different cohorts, which increases confidence. And as a result, I can show you that siRNA log fold change, this differentially expressed gene list I get from depletion of A5, upregulated genes, downregulated genes, indeed split the primary and secondary targets. And secondary are bad risk, and then primary are good risk. And there is a relationship with A5. So according to this, we are finding that A5 could be really important in determining survival of a tumor, and it has a strong association with ER status. And we further done several more analyses I will just briefly mention. And remember, estrogen receptor is inducing certain genes, some of which are transcription factors, and E2F1 is one of them. And also E2F1 is like at least a tertiary signal that is induced by estrogen receptor. So E2F1 is interesting because A5 has binding sites for E2F1. So E2F1 could be the factor that is regulating A5 as a secondary target, making it as a secondary target. E2F1 and RB1, which we see that depletion of A5 really reduces the phospho-RB. So RB and E2F1 bind together. If they are together, E2F1 cannot be inducing cell cycle. So RB needs to be bound to E2F1 and not phosphorylated so that it is bound to E2F1. That means cell cycle arrest. If E2F1 is free, it can go bind to the DNA, induces cell cycle genes, including A5. So RB and E2F1 is negatively correlated. This is a meta-analysis. It's known as forced plot, you might know. These are different GSC numbers, and each one is a data set from GEO. They're different data sets. The x-axis shows the correlation between RB and E2F1, and what we show that RB and E2F1 are negatively correlated. So they're all on this side of the graph, suggesting that in all of the data sets I'm looking at as a meta-analysis, they're all downregulated, which is what I expect, right? E2F1 is high, cell cycle is high. E2F1 is low, cell cycle is low, and it's just the opposite with RB. But this is more interesting. So when I look at RB expression, right, together with the A5 expression, I still see some form of negative association. So that is reminiscent of estrogen receptor. But what is happening if RB is depleted? A5 is very high. If RB is depleted, E2F1 is very high. If E2F1 is free, it can induce A5. And those cells or those patients with increased A5, decreased RB are shallow deletion. So they really don't have much RB because of copy number alteration, and they are negative, ER negative. So RB could be one of the solutions. Then RB and E2F could be explaining why we have A5 high in ER negative. And because RB is not present, E2F1 is there and A5 is there. So this could be, and it still needs to be shown. And this is our next study. We also looked at another ChIP-seq data. In this case, this data is very interesting because it shows that A5 has a promoter for E2F1. This has been shown by a ChIP-seq. But if you deplete RB, by shRNA, and this binding to this site of A5 by E2F1 increases. So a bigger box means that there's more E2F1 bound when there's no RB. So this also shows another layer of information suggesting that A5 promoter can be bound by E2F1, which can be regulated by RB. So overall, this is our working model right now. And we have the A5 present on the membrane together with A3 and B4. And acetylcholine may act on it, bring the calcium in. Calcium increases more calcium through the endoplasmic reticulum. This increases AKT, MAPK, etc., and estrogen signaling. And progesterone is released, but E2F1 also is released. E2F1 goes back in, induces A5, and the cycle keeps going on. So this is our hypothesis. And if we deplete A5 when there is not much A5, it is not going to be present at the membrane. So something else will happen here, maybe less calcium or some other parameters will work on, and it will limit estrogen receptor signaling. But it will limit only those that are producing E2F1 secondary targets. Because what we see is primary targets, progesterone is increased. I didn't mention this to you, but this is also what we have seen. Progesterone is one of the factors induced by A5. So something is happening to the estrogen signaling. Primary and secondary targets are splitting, and that splitting is reducing A5 further, and this will lead the cell to go through cell cycle arrest. If there's estrogen receptor, if there's estrogen signaling, and if there's P53. So what are we doing next? We are looking for novel compounds to identify similar profiles to A5. And I'm just going to show you this particular clustergram here. Again, we have data from different data sets. Estrogen provided, estrogen given, MCF7, tamoxifen, lazoxifen, IC fulvestrant, etc. These are known inhibitors of estrogen receptor signaling. This is estrogen receptor inducer, E2. This is amino acid depletion. The cell has no amino acids here. Dioxin is aryl hydrocarbon agonist. So these are like different treatments. All of this data come from GEO. And these 5 molecules, 53, 51, 50, 49, and 48, we have, in collaboration with Ankara University, uh, designed and synthesized these. These are novel compounds. So what we're finding is that novel compounds are acting similar to the estrogen receptor inhibitors, and they are inversely correlated with estrogen. And when I compare this with A5, these novel profiles and known profiles, this shows that negative association with the inhibitors, sorry, positive association with the inhibitors, positive correlation, negative correlation with E2. So what I'm showing you is that we're using cluster analysis and comparative transcriptomics to find similar chemical profiles to A5 depletion and estrogen receptor modification and modulation. So I want to end here. I think, let me just check my time. I have enough.
We
Also
have
do in
enough
vivo
time for
studies,
questions as well.
but I'm not going to talk about the in vivo. So we have come up with this A5 depletion as a strategy to deplete cell cycles, to deplete A5 and result in cell cycle arrest. And we're trying to find out how this is and why this is happening. And we are trying to find this by using bioinformatics together with all kinds of different comparative transcriptomic analyses. So I'm going to come back to here and answer your questions if you have any.
Okay, thank you so much for your this nice explanatory presentation. Now we
Thank
can
you.
have your questions in chat box. In the
So,
meantime, actually, I have one question.
yes, uh-huh.
You mentioned in the experimental part doxorubicin and siRNA in a synergetic,
Yes, manner,
yeah,
yeah. Mhm.
in a, they are working in a synergetic manner. I wonder, have you tried doxorubicin with siRNA in a healthy cell line?
Mhm. Very good question. We haven't tried it. We have tried it with um, ER positive, ER negative cells, and it is working in both of them. But this would be very important. Like if doxorubicin is killing uh, healthy cells, which it does to a certain degree, but together with
Yeah,
SI, if it just is effective as cancer cell, this would be bad for the patient, right? So it cannot be a good
yeah.
strategy. I think
But
we need
still,
to look that up.
very
Mhm.
important data, I think, because now you can put doxorubicin in a liposome. We have different targeting strategies. So as
I
long as
agree.
it works in cancer cells, so it's really great. And now
This
we
needs to be
have,
done. This needs to be done. You're very right, and I think that's one thing that is important.
yeah,
Mhm. I mean, it
we
would
have
be
one
nice if it
question.
like changes and it makes it even like you don't see the same effect in cancer, non-cancer cell line. That would be nice.
Evet.
So the problem, of course, is to find a normal cell line in. So you can use these MCF10A such data to see that if they're not exactly normal, but they're transformed and they're not like they don't make cancers when they're transplanted. So
Yeah.
that would be interesting to look at.
Okay. We have
Mm-hmm.
one question right now. Can you see the chat
Yeah, I
box?
can see. I'll go over and thank you for the question. How you actually decided to work on this and cancer, apart from the gastric cancer and cholinergic receptors? Okay, this is a good question because it tells you the power of bioinformatics. So my background is cholinergic receptors, but I worked with them in brain. So I was interested to see if any cholinergic receptor is expressed, and this is happening in like beginning of 2010 or something. It's quite old. And so if they were expressed in cancer, so at that time there was no TCGA, there was there wasn't that much data, but there was SAGE expression analysis. SAGE was another method. And by doing a SAGE analysis, I wrote a proposal to Tubitak, and I identified A5 being highly expressed in several cancers. At that time, even A5 was not known. So this is how we started to profile cholinergic receptors in breast cancer. And then my next project was this SI actually. So initially we have identified that cholinergic receptors are expressed. And so do you think it is advantageous to work in a lab where wet lab and bioinformatics parts are going together? I 100% I think so. But if you can be completely in a bioinformatics lab, and the bioinformatics lab is so huge, so much resources, and a lot of people to interact and collaborate, then you don't need your own lab at all. If you're able to collaborate with groups that are found next to you and to get actual data that is like fresh new data, so that you can go ahead and you have a hypothesis, you test it, and that is most fruitful. And one other question: cancer is very heterogeneous. Um, yes, it is. What do you think about studying these in a single cell level? And that is really, really what things are going, right? You probably asked this because of that, because now that there's single cell sequencing, so you can take even the cancer cell, treated, untreated, or put them together, label them differently, look at them, how many different populations emerge, and having this single cell equipment in your lab may not be always possible. So that's the limiting factor. But as soon as those single cell machines go into the lab, now the sequencing is so easy. You get your RNA, you send it, they sequence it. But if you really don't have your single cell sequencing machine, you may have a little bit problem in preparation. But once you solve this, I think that's the next thing to do, and it would add so much more. Thank you for your input about the interdisciplinary work. I have to say that this is really the key, I think, interdisciplinary. Let it be like biology and bioinformatics or different parts of bioinformatics like proteomics, phosphoproteomics, metabolomics. You may not be able to do everything in your own lab, but you can, even bioinformatically, but you can always go interdisciplinary. Have you considered it as a therapeutic target, cholinergic signals? Do you think it can be a promising target? So it is actually, cholinergic signaling is already a target, right? Why? For example, Alzheimer's disease. You know, Alzheimer's disease, you're too young, so you might not have encountered it. But I have encountered actually personally around me, but also from the literature, there's a treatment for Alzheimer, which does not prevent Alzheimer. But when someone is going through the Alzheimer, on the road to Alzheimer, they give him AKE inhibitors. AKE is acetylcholinesterase. So remember the first figure I have shown you, like there's AKE in the presynaptic cleft, and that acetylcholine is produced by the cell excreted, and there's an enzyme called acetylcholinesterase that degrades that acetylcholine. Otherwise, acetylcholine will constantly be present, and it's going to bind to the nicotinic receptor, and it's going to make that nicotinic receptor paralyze person as well, kill the person. So you cannot have too much acetylcholine. There is an enzyme that degrades it. And that enzyme, acetylcholinesterase, can be inhibited to a therapeutic level. So that what happens is there's a little bit more acetylcholine than normal, and that triggers the neuronal cells, which makes them more active. And Alzheimer's disease patients are currently using this drug for years. They start at the very early on, and it keeps the disease pretty stable. So that is one approach. You modulate the ligand, the acetylcholine, by enzymes or itself, and you either increase or decrease the signal. The other one is cholinergic receptor. And there is also in treatment a cholinergic receptor. And I hope you're not smoking, but if you're smoking, there is a drug that targets cholinergic receptors, and that can actually prevent the cholinergic signals going into the cell, which makes the person desire less nicotine, because nicotine and acetylcholine do exactly the same thing. Nicotine binds to the acetylcholine receptor and induces. So for cancer, it hasn't been a target. So that's why it's interesting. So cholinergic signaling is a very good target for myasthenia gravis, so muscle disease, for brain diseases, etc., but not for cancer that much. So it could be a good target. It's getting attention.
Yes.
Very nice question. Thank you.
I guess we don't have another question right now.
Okay, thank you so
S-
much.
So I would like to thank you on behalf of RSG Turkey. That was a very really nice talk. And generally, we are focusing on the bioinformatic part of studies, but it's also good to know the experimental parts, the
So,
wet lab part of it.
so you're all students, right? Are you
Yeah,
all
mostly.
students, early researchers?
Uh, Actually, it's a mix. We have undergrad students, we have PhDs, postdocs.
Uh-huh.
It's all together.
Very good, very good. And mostly bioinformaticians, right? Not like some of you are doing both, I assume.
Yeah, but mostly bioinformaticians.
Okay. Very good. I'm glad that I could give you a different point of view, and it is so important to have you all with us. And like my lab does both. So I have students who work with bioinformatics and in connection with what we find in the lab, and everybody is intermingled. So we actually do, some people do both, some people just do one because the preferences vary. And we find it very, very fruitful. So I wish you all in your careers best and healthy days and healthy careers. And thanks for
Thank
inviting me.
you. Thank you so much again.
Bye.
I think we can now close.
Thank you. Thank you so much. And you
And
say
I would
bye.
like to thank our attendees for their participation. As I said, you can follow us on social media on Instagram, LinkedIn, Twitter, and Facebook to hear about more about our next events. Thank you so much. We are done for today.